All right, so we just finished up our animal cells and our plant cells. Those were considered eukaryotic cells. They did have a nucleus, they had membrane-bound organelles, they had DNA that was found in the nucleus, and we are now going to switch gears and look at prokaryotic cells, which you're going to see look much different than eukaryotic cells. First thing I do want to talk about before we solely talk about prokaryotic cells is to compare and contrast the eukaryotic cells, so your animal and plant cells, versus our prokaryotic cells, which are our bacteria cells. The first thing is a prokaryotic cell does not have a nucleus, but a eukaryotic cell does. So if you remember, both plant and animal cells always had that nucleus, usually near the center of the cell. When we look at a prokaryotic cell, you will not find that. Prokaryotic cells do not have a nuclear membrane. Eukaryotic cells do. Remember, nuclear means that it's going to be surrounding the nucleus, so if there is no nucleus present, there's not going to be a nuclear membrane. With a eukaryotic cell, something does need to protect that nucleus, so you do have a nuclear membrane. This was also called a nuclear envelope. For the DNA, or the genetic material, both prokaryotic cells and eukaryotic cells have DNA. It just comes in different forms and looks different in the cell. So for a prokaryotic cell, it kind of looks like a big ball of yarn in the middle. It's circular and it is all kind of wrapped up. For a eukaryotic cell, you do not physically see the DNA because it is housed within the nucleus, but when you look inside that nucleus, they are paired up, they are linear chromosomes, and it's a little bit more organized than what you would find in a prokaryotic cell. So for examples, prokaryotic cells are going to be bacteria cells. We're going to continue with bacteria cells for about the next week or so. And we just finished up our eukaryotic cells, which were the plant and the animal cells. So looking at our prokaryotic cell and our eukaryotic cell, the first thing you can see is that they look very, very different. This almost looks like a pill, and this is very round. This, once again, is an animal cell over here with the round shape, but it is more specifically a eukaryotic cell because it does have a nucleus. The prokaryotic cell, you can see, does not have a nucleus, but it does have that DNA here in the middle, and we call that a nucleoid, and up on here it's labeled as the nucleoid region. You can also see one of the big differences is there are very few organelles in here. You've got your nucleoid and you have some of these dots here that we call ribosomes, and when we look over here, we have all of our organelles that we have just studied. I'm not going to go through them all, but there are several more than what we have over here. The last thing I want to point out that is similar is the flagella. You can see that these whip-like structures are found on a prokaryotic cell, and they can also be found on a eukaryotic cell. These hair-like structures are not considered cilia. We'll talk about these more when we look at the structure. But things they have in common: they both have a plasma membrane or a cell membrane. They have a cytoplasm, and then they also have that genetic material, which is DNA. So the first thing I want to talk about is how are prokaryotic cells classified. The first thing I want to mention is that the classification has changed. So when they were first classified, they were put into this kingdom called Monera, and this meant that they lacked nucleus and they also did not have membrane-bound organelles. They have a cytoplasm, but inside that cytoplasm we just have that nucleoid, which is that DNA, and ribosomes, and no nucleus. However, now that we know more about prokaryotic cells, they have been divided or classified a little bit differently. We now have something called a domain, and this is a higher classification than a kingdom. And the two domains are going to be domain bacteria and domain archaea. Domain bacteria, these are going to be the bacteria cells that we most commonly see. These are the ones that are going to affect humans. If you hear a doctor or somebody talk about bacteria, it's most likely going to be in that domain bacteria. The domain archaea bacteria are going to be found in more extreme environments, deep sea volcanic vents, very hot sulfur springs, places that you wouldn't think things could survive, but they have found this domain archaea bacteria there. So we're going to focus today on the domain bacteria, the ones that we find everywhere, just to remind you that this is the bacteria we do find in our everyday environments. When we're looking at the classification underneath the domain, we're going to look at the kingdom, and this kingdom is titled Eubacteria. There will be a lot of times where you see that it just calls it kingdom bacteria. It means Eubacteria, same thing. Once again, they are found absolutely everywhere, but not in extreme environments. That's where we find archaea bacteria. And they are going to have strong cell walls with this thing called peptidoglycan. And this peptidoglycan is made up of sugars and amino acids, which we will learn more about later in the notes. So the first thing I want to talk about with bacteria is the shapes that they come in. We have three main shapes. We have cocci, which means round or spherical. We have bacilli, which means rod-shaped. And then we have spirilla, which is a spiral-shaped bacteria cell. In your chart on your notes, I want you to go ahead and draw these kind of basic shapes. So underneath cocci, you want to go ahead and draw a circle to show that it's round. Underneath bacilli, you kind of want to draw this rod-shaped, it kind of looks like almost like a pill. And then lastly, for the spirilla, I want you to draw kind of that wavy line to show that that means spirals. At this time, you probably want to pause this so that you can draw, and I'm going to continue moving on with the notes. Okay, so underneath each of the different shapes of bacteria, we do have different arrangements of those shapes. The first one we're going to look at is cocci. Remember, those are circular. The first one is coccus. That is one single cocci bacteria, so one single round. The next one we have is diplococci. The di means two, and the cocci means round, so we have two round bacteria cells. Staphylococci is going to be a bunch of round bacteria cells. The word staph actually means a bunch, and it kind of looks like a bunch of grapes. Streptococci is going to be a chain of round bacteria cells. So that word strep actually means chain. And lastly, we have something called a tetrad, and tetra means four, so you have four round bacteria cells grouped together. You do need to draw these in your notes, so this would be a good place to pause and draw these in while I continue with the notes. Our next arrangements we're going to look at are going to be of the bacilli. Remember, these are your rod-shaped bacteria cells. The first one is Cocobacillus. This one is actually going to be a mixture of kind of the round bacteria cell with the more rod-shaped, and it kind of makes that oval shape. The next one is just what we would say bacilli, that is one single rod shape. Diplobacilli means two rods together, so remember di means two. And then we have streptobacilli, which means a chain of the rod shape. Remember, strep means chain. We could also have staphylobacilli. That would just mean there's a group of them, but these are the main ones that I would like you to know. So this would be a good place for you to pause again to go ahead and draw these in your notes. The last one we're looking at are spirilla. These I just put pictures in your notes because it's kind of hard to draw the difference between the spirillum and the spirochete. The biggest difference is the spirillum is going to be a thicker spiral bacteria, and the spirochete is going to be really, really thin. The way that I look at it is the spirillum is kind of like a big fat spaghetti noodle, and the spirochete is more of like the really thin angel hair pasta. Last thing I'm going to talk about in these video notes is how do bacteria actually metabolize. So remember, that's how they obtain energy, thinking back to those 7 characteristics of life. We have 3 different types. So the first type are going to be called heterotrophs. The word heterotroph actually means other feeder, so hetero other, and the troph is the feeder. This means that they have to get food from some other source. So they cannot make their own food, or they cannot synthesize their own food. So they have to go around and find other organic nutrients around the environment or even off of other organisms. So When we're talking about other organisms, sometimes that means that there are going to be dead animals or dead organisms that are being decomposed by heterotroph bacteria or prokaryotes. We will call these saprotrophs or saprobes. And so when you see like a dead or decaying animal on the side of the road, most likely you're going to have some type of saprotroph or saprobes actually helping to decompose it. So as the flesh is kind of disappearing, what's really happening is that you have prokaryotes or bacteria cells actually eating away at that dead organism, and then before you know it, you have a pile of bones. Our next one that we want to talk about are called photoautotrophs, and the word photo means light and autotroph means self-feeder. So they usually carry out photosynthesis. You can see in this picture they actually have green pigment, and that green pigment is chlorophyll, so very similar to what is found inside of a plant cell. So they're going to use inorganic molecules, so most specifically carbon dioxide they'll pull out of either the air or the water. They will also need water, so H2O they will need to help go through photosynthesis. The last thing they need is also sunlight, so they cannot carry out photosynthesis without that sunlight. But these photoautotrophs will make their own food. They do not need to find their food source anywhere else. My example for this is called cyanobacteria. This is frequently found in like water areas, ponds, places where there's really still water, and it kind of almost looks like if there's enough of it, can look more like algae. The last one I want to talk about are chemoautotrophs, and this means chemical self-feeder. So the chemo means chemical, and once again that autotroph means self-feeder. So these also do not need to go find food. They will make their own food. They do rely on inorganic compounds, most specifically containing either nitrogen, which is the N, or sulfur, which is the S. And they do not go through photosynthesis. They actually go through something called chemosynthesis. So remember, photo meant light, so photosynthesis means you need to have the sun. Whereas chemo, remember, means chemical, so chemosynthesis means you need to have specific chemicals in order to make your own food. One of the most specific, most well-known bacteria that is a chemoautotroph will be the nitrogen-fixing bacteria, and that is part of the nitrogen cycle, so it will help absorb nitrogen or break down nitrogen that will get put back into the nitrogen cycle. This is where we are going to end with the notes. We will continue doing these notes in class over the next several days. Please let me know if you have any questions in class tomorrow.